All right. I want to say a very special welcome to Andy Dell, and I will ask you to present yourself. I hope that you will all enjoy tonight's seminar. So over to you, Andy.
Thanks, Linnea, and thanks for the invitation to speak to you all. It's great to see we've got well over 100 people participating in the webinar at the moment. What I'd like to do tonight is to talk through the problems that we have with breeding within closed stud books. My background is that I've been heavily involved with horses for over 60 years and with Cleveland Bay horses for the best part of 30 years. And my experience with Cleveland Bay horses and my background as a biology teacher led me to look at the way that we breed these rare animals and to think about, well, if these animals are really rare, what do they actually do with the rarest of the rare animals? And that brings you to look at the zoo world and what they do with the likes of giant pandas, Siberian tigers. And in studying that science and my involvement with the Cleveland Bay Horse Society, we've actually managed to put in place a breeding program that I'll talk about later that's had considerable success in stopping the loss of genetic diversity within that breed. But to backpedal slightly, what I want to do tonight is to talk about the problems that breeding within those stud books actually have to deal with, to look at strategies that we can put in place to mitigate against those problems, and also to look at how we can monitor our rare breeds and how we can secure them into the future. So I think whether we're breeding Cleveland Bay horses, Suffolk Punches, or Shetland ponies, there's an awful lot in common. And hopefully there are some things that you can take away from this meeting that will make you think about how you run your breeding programs. I am involved with the Rare Breeds Survival Trust here in the UK. I'm a member of their expert advisory panel, but the Rare Breeds Survival Trust every year produces a watch list of all of our rare types of livestock, be that sheep, cattle, pigs, poultry, or the equine breeds. And they monitor them to report to our government, the Department for the Environment, Food and Rural Affairs, and also to our Farm Animal Genetic Resources Committee. And the reason that they report to those bodies is because all governments are actually tasked with reporting their biodiversity to the Food and Agricultural Organization of the United Nations, whether that is plant biodiversity, wildlife biodiversity, wild animal biodiversity, or whether it's livestock. And so we carefully monitor our livestock breeds, and you'll see that in the equine list, the Cleveland Bay horse is right at the top of the breeds that are seen as priority breeds because they've been genetically compromised and continue to be genetically compromised. And we share that classification with the Dales pony, the Dartmoor pony, the Eriskay pony, the Exmoor, the Hackney horse, and the Suffolk horse. And there are other breeds down there that you'll probably recognize as well, but down at the bottom of the list in terms of UK native breeds that they're monitoring the Shetland pony, they're also monitoring the Irish pony and the Irish draft horse. So there are breeds there that you will recognize, and your own breed, the Shetland breed or the Shetland pony, or certainly the UK population of the Shetland pony is there in that watch list. So whether it's Shetland ponies or in this case Eriskay ponies, all of our breeds within small populations have inherent problems. And that first and foremost is that if you maintain a small population within a closed stud book, in other words, you set up a stud book and you put a ring fence around it and you say that nothing else from outside in terms of genetics is going to come in, then it will start to lose diversity. There's nothing that you can do about that. It will happen. Maybe what you can do is control how fast it drifts away, but it will lose diversity much quicker than a large population. And I'm going to talk in a moment about the mechanism by which that happens, and that's genetic drift. And genetic drift actually means that you lose variation, the types and the number of different genes that your breed is carrying. And associated with genetic drift and breeding within closed stud books is inbreeding. And that in turn can bring about what we call inbreeding depression. And I'm going to talk about that in a little bit more detail in a moment. So what do we mean by genetic diversity? We know an awful lot about the genetic makeup of the horse now. The whole genome of the horse has been sequenced. It carries over 2.7 billion genes. And if we could take all of those genes and put them in a big heap, and we represented them by coloured sweets, there would be one huge great pile of coloured sweets. But we'd find that in amongst that pile, there would be more of one colour than of another. And if, for example, I choose 5 of those colours and that's our heap, we can see we've got a lot more red sweets than we have green and blue sweets. The green and blue sweets are the rarest in the population. And if we think back to the genetics that we learned in biology lessons at school, we know that as mammals, we are what we call diploid. We carry 2 copies, 2 alleles at every gene. And so when we're breeding, if we bring our dam and our sire to the table and we have a look at the dam here is carrying genes A and B for whatever trait it might be. And at the same place on the DNA, the sire is carrying C and is carrying D. And those animals are completely unrelated. If we cross those animals, we're going to cross them twice. So let's have a look at the first cross. We cross this dam and this sire to breed this filly foal. It's actually inherited the gene A from the dam, and it's inherited the gene D from the sire. So at this particular gene, it's carrying A, D. Let's do the same cross, same dam, same sire, and the same 4 genes that could possibly be passed across. In this case, on the second cross, the dam has passed across B, and the sire again has passed D. At the top, we had A, B, C, and D all present. But in 2 crosses, we've now got A, B, we haven't got C, we've lost it, and we've got 2 copies of D. So C has been lost by genetic drift in those 2 crosses. And that was just looking at one gene within the population. Bear in mind that when we make those crosses, 2.7 billion genes are being crossed and are being sorted. So if you scale that up, you can imagine the huge loss of diversity that could result. Keeping the same analogy of our coloured sweets, if we start with 10 different colours in this dish and we make those crosses, we did 2 in the illustration before, let's do quite a lot more, however many that might be. But if we sample after quite a lot of crosses, now we've got the same number of genes, there's the same number of sweets in the tray, but only 6 of the original colour variants are left. And if we carry on doing that experiment in this tray, a lot more crosses later, we've still got the same number of sweets, but now there are only 2 different types of sweet left, and there's only 1 of the blue, but everything else is red. So we've lost an awful lot of variation. Even though we've got the same number of genes, we haven't got the variation that was in the original population. So that's, in simple terms, genetic drift. What I want to do now is to look at it in equine terms, and certainly Most of our rare breeds in this country, when we register them and issue passports, we do that based on parentage verification. And parentage verification is done by DNA analysis. And to be more precise, it's done by, at the moment, what we call microsatellite DNA analysis, in which we pull a hair sample, or a vet takes a blood sample, it's sent to a laboratory, and it's tested against a panel of 17 markers that are approved by the International Society for Animal Genetics. And some of you may know that there are moves afoot to move to a more advanced type of marker in a different type of panel, and that will be what we call SNPs. But at the moment, we're using microsatellites, and there are 17. In the future, we may use a lot more markers, not just 17 markers, but possibly 70,000 markers. But that's 2 or 3 years down the road. At the moment, this is the sort of report that a breed society would get back from the laboratory. These are the various markers that it's being typed against. To make things easier to understand, I've taken the data on that printout and I've translated it into this table. These are the markers, VHL20, HTG4, down through to LEX33. And there are 17 markers in the approved panel. And given information from the sire and given information from the dam, we can say whether the progeny is actually progeny of that mating. So for example, the sire at VHL20 is carrying IM, the dam is carrying IP, and the progeny has been tested as carrying MP. Well, is that possible? Well, yes, it could have inherited. M has clearly come from the sire, and P has clearly come from the dam. And if we work our way down through that list, each of these checks out. If we just do a random check here, the progeny is carrying KK at ASB2. Well, it needs to find a copy of K from the sire and from the dam. Can it do that? Yes, it can, because the sire was carrying K there. The dam was carrying 2 copies of K. That's what we call homozygous. Homozygous meaning it's got no variation at that particular gene. It's carrying 2 copies of the same. So it's quite possible. And in fact, this was a real mating. It was real progeny, and that progeny, the parentage was verified, and it went on to be registered in the stud book as being purebred. So that's when things go according to plan. Here's another example. And this time, if you look at the report, it says DNA excluded. Well, what does that mean? Again, it's the same panel of markers. This is a different mating. I've drawn up the same sort of table. We've got the same markers down the side, a different sire and a different dam, but let's look at the results. First of all, at the very first marker, the progeny is carrying N and P. Well, where did it get N from? Could have come from the sire. It could have come from the dam. Where did it get P from? Well, it certainly didn't inherit P from this sire, and it's impossible for it to have inherited it from this dam. And if we work our way down this list, you'll see that there are a few markers that don't work out. Would this one work out? The second one, it's KM. Well, yes, it could have got K from the dam, and it could have got M from the sire. But if we carry on down the list, here's another one to check. It's KO. Well, yes, it could have inherited O from either of the parents, but K wasn't represented in the parents. It's impossible. So they had to go back to the breeder and go, this isn't right. What are the other options? And do some more DNA checks before they verified what the parentage actually was. So that one wasn't registered with that parentage. So that's the background to how the system works. What I want to show you next is another mating. This is a real mating of animals that were very highly related to each other. The sire and the dam were very highly related, and I want to show you the consequences of that. In the same way, to make it easier to understand, I've tabulated the results. And first of all, I want to look down the column and see what the sire was carrying and see where the sire was homozygous. In other words, it was carrying identical copies. So here it's carrying 2 copies of N. It's carrying 2 copies of M. Carry on down through the list. There's 2 copies of I there, another 2 copies of I there, and 2 copies of L. So altogether, the sire was homozygous at 5 out of the 17 markers. Let's have a look at the dam. That's not actually that abnormal within Cleveland Bays to be homozygous at 5. The dam, she was a homozygous at 1, 2, 3, recorded as 3. These are question marks. This was a very early test and wasn't tested at all the markers. So we know she was homozygous at 3. So the sire was homozygous at 5. The dam was homozygous at 3, neither of which is abnormal. Now let's have a look at the progeny. We know that the progeny is a very highly inbred animal. It's homozygous at 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So that shows you how suddenly an inbred animal can become very, very homozygous.
We have a question from the chat.
Sure.
Is a de novo mutation possible if it's only in one instance where the markers doesn't match?
Can you say that again?
Ask
the question again.
Yeah, is a de novo mutation possible if it's only in one instance where the markers doesn't match?
Right. In terms of parentage verification, if it's a mismatch at one site, they probably go away and retest. And that's part of the reason they want to move away from the panel of microsatellite markers to the panel of SNP markers, where they're not using just 17, but 70,000 markers, of which they won't use all 70 for parentage verification, but it provides us with a much more accurate picture. But at the moment, a mismatch at one site would just, it wouldn't preclude registration, but they would probably go away and do some more testing. Okay.
Excellent.
So in terms of inbreeding, an animal is inbred if its mother and its father share a common ancestor. So in other words, if we draw out the pedigree, and whether that's at grandparent level or way back deep in the pedigree, sharing a common founder, if it's carrying genetics from that animal down both sides of the pedigree, it's inbred. And back in 1922, a geneticist called Sewell Wright came up with the idea of the concept of an inbreeding coefficient, which measures the degree of inbreeding. In other words, how many of these genes are identical by descent down through a pedigree. And to put things in perspective, we certainly don't go away and marry our cousins, certainly not in the UK and probably not in Sweden either. The reason for that being obvious, and at cousin level, a progeny of a cousin mating would be 12.5% inbred. A brother-sister full sibling mating would be 25% inbred, and a mating of a sire with his own female progeny would be 25% inbred. So in other words, 12.5% is the equivalent of full cousin mating, and 25% is equivalent of full sibling mating. And Cleveland Bay horses, average inbreeding in Cleveland Bay horses has already reached over 22%. So we've got quite a problem. And I'll talk to you about what research tells us that the level of inbreeding, certainly within the UK Shetland pony population, is later on. So let's just look at the genetics of inbreeding again, go back to our diagram of our dam sire mating. These animals here were unrelated. And in that cross, the same cross that we had before, the progeny are unrelated in that first generation. But if we take these two progeny and then go on to mate them, they share common ancestors, because share common parents. So the offspring is inbred, and that's obviously very close mating. In a very shallow pedigree, you can expand that right back to 32 generations and common founders and find very deep inbreeding within breeds. So does inbreeding matter? Yes, it does. It reduces gene diversity, and I've shown you that in real examples from parentage testing. And in turn, what it does, it reduces general fitness. And I don't mean physical fitness, how fast your horse can go. I mean how fit your horse is for breeding, how easy it is to get in foal, how well a horse can adapt to environmental challenges. And above all, what increasing levels of inbreeding do is they greatly increase the risk of what we call undesirable genes or deleterious alleles being expressed. And these are genetics for traits like in the Fell pony and the Dell's pony. Over here in the UK, we have lethal foal syndrome, and there are plenty of other mutations that we can talk about that cause lethal traits within horses, certainly associated with the Overo colour. And there are also deleterious mutations within Shetland ponies and miniature ponies that I'll talk about in a moment. But they're certainly associated with increasing frequency as the breeds become more inbred. In fact, recent studies have shown that out of the 2.7 billion genes that make up the whole genome of the horse, now it's been completely sequenced, there are 46,000 potential deleterious alleles that have now been detected within the equine genome. They're not all lethal, but what they do know is that some are lethal at embryonic level, and there is recent work that is shown that it's actually the presence of some of these deleterious traits that are making it much harder for our rare breeds to get in foal, because in fact what is happening is they are getting in foal, but the traits are lethal at embryonic level, and they're actually being absorbed or aborted very early on in the pregnancy. I talked about very deep inbreeding, and certainly within populations like the thoroughbred horse, we know we can trace that back easily 36 generations. The Cleveland Bay horse, we can trace back in some cases 36 generations, and we know that there are common founders across that baseline of the population, which are certainly present on both the male and the female side of the population, and that they are the reason for a lot of our very deep inbreeding. So we know a lot about Cleveland Bays, but we also know a lot about the Shetland pony. And just in scanning through the literature, I came across this paper by Schiernick and al. 2019, so it's relatively recent. This is not just from pedigree analysis, but this is from DNA analysis, and it's DNA analysis not from just the microsatellite parentage testing, but from SNPs testing, where they are testing 70,000 markers. We can measure inbreeding from the pedigree or from what we call runs of homozygosity, and I'll expand on that later. But if we look at some of these breeds, it's well publicized that the Frisian horse is one of the most inbred of all of our breeds. In this case, from pedigree, they detected that the average inbreeding was 25%. I've already mentioned that a full sibling mating is 25%. That's the level that they're at, and they were detecting a maximum of 33%. I wouldn't be at all surprised if they looked deeply within the stud book, they'd find some examples higher than that. That wouldn't be unusual. And in terms of molecular data, actually going and looking at the SNPs panel, then they're finding a mean inbreeding slightly lower than from the pedigree, but it definitely is 22%. And they found a maximum when they looked at the SNP samples of 30%. And that's looking at a sample from 1,485 animals. That's a very representative sample. Looking at the the Netherlands, this is the Netherlands Shetland pony population. They've looked at over 900 Shetland ponies. They've looked at the stud book and at DNA analysis. They found that the mean inbreeding within the Shetland pony was 17%. That's already up above cousin mating and on the way towards sibling mating. With a maximum of 36%. In fact, if you look at this panel from the stud book, it shows at the highest level. And in molecular terms, nothing like as high as the Frisian, but actually the second highest breed of the ones that they've studied in that list. And with a maximum there of 33%, which is pretty much validating what they were finding from pedigree analysis. And that was from still a representative sample of almost 1,000 ponies. So that's from the Netherlands.
Can I ask?
Of course you
can. A percentage of 35% or 33%.
Shall I go back one?
Yeah.
I'll just go back one.
So 25% is sibling mating. What does
full sibling mating, brother, sister,
yeah.
or a sire with his own filly foal?
So yeah. How can it be 36%, or is it the
right? Okay, so you can actually be go even more inbred, because if you've got inbreeding that isn't just from the parents, but it's from deep within the founders as well, you can get inbreeding well in excess of that. In fact, you can get inbreeding going right up to 100%, where all of your genetics on both sides of your pedigree are completely related to themselves. So an inbreeding coefficient of in excess of 25% is easily possible if the parents are already related to themselves from some level within the pedigree. And if I go on to have a look at how we measure genetic inbreeding from runs of homozygosity, if you remember back to where we looked at the parentage testing that I showed you how we ended up carrying 2 copies of the same gene at a marker. Well, we can actually find long chains of that inbreeding going on through adjacent markers in this, what we call the SNPs panel, the single nucleotide polymorphisms, if you want the full term. But those chains can be very short chains, medium length chains, or long chains. And what they found is that the short chains belong to relatively shallow inbreeding in the pedigree, and the long chains relate to very deep-seated inbreeding. Okay, so we can actually tell from molecular analysis whether it's deep inbreeding or whether it's shallow inbreeding that's causing that inbreeding. And if we look at the inbreeding within the Frisian horse, we can see that there's a chunk from relatively recent inbreeding and a chunk that's deep-seated within the pedigree. And it comes out in a similar sort of pattern, although not quite to the same scale, within the Netherlands Shetland pony as well. Okay, so this isn't just something that we've worked out from looking at pedigrees, which are statistical analysis. This is actually from molecular analysis from looking at the panel of SNPs markers. This model.
I also have a question.
Sure.
From the last picture, when you talked about the Frisians.
Let's see if I can get back to it for you. No, we're going the wrong way. Let's just jump back one. Let me just see if the technology might beat us. Right, here we go. Is that the picture?
No, it's the one before,
the one before that. Let me just go back one more. If it'll let me. There we go.
Yeah. And this, it says mean. Is that like the
average?
The average
for the sample. So in the case of the Frisian horse, they've sampled 1,485 animals. They've looked at the pedigree of those animals and the stud book data, and they've also looked at blood or hair samples from them. And in the pedigree analysis, they found that the average inbreeding is just over 25%. the least they found, the minimum was 18%, and the maximum they found was 33%, but the mean, the average was 25. And then in terms of molecular analysis, the mean they found was 22%, which is slightly less than what they found from the pedigree. And similarly, the maximum was 30%, which again is slightly less than they found from the pedigree. But what you've got to bear in mind is if you do pedigree analysis, you're relying on how reliable and how accurate the stud book data is, and it's subject to any human errors, either recent from recording it or digitizing it, or what did our ancestors say, you know, record when they started the stud book, you know, were the pedigrees that they recorded accurate? But we can move away from that with molecular analysis when we actually start to look at the SNPs panel. This doesn't lie. This is actually what we're finding in that animal in molecular terms. But interestingly, the two aren't desperately far apart in the Frisian horse. So
no, but the sample must be very high percentage, because now the Frisian horse is at 17.8 as the whole population of the.
Right. Well, this was from a sample of the Netherlands Frisian, and there is
the Netherlands, 17.8.
And if we could have a private conversation later rather than hijacking the seminar for people.
No, I'm just wondering about the.
I'd be interested to know what paper it is you're getting your figures from. Perhaps we can talk later. Okay.
Okay, thank you.
Thanks. So just to move on, this is a model called the Extinction Vortex, and it's an analogy that's used for captive wildlife, for wildlife populations out there in the big wide world, but also for livestock populations. And if you start off with a small population, and especially our ring fence populations, and you inevitably inbreed from them, there will be random genetic drift, which will lead to a loss of genetic variability, a loss of genetic diversity using the models that we already looked at. And that will reduce the fitness and the ability of the population to adapt to future stresses and strains. We know it also increases the frequency of lethal variants, which will lead to a lower level of reproduction, to higher mortality, and that takes us around a spiral, which leads to an even smaller population. And you can just see that if we carry on going around that circle, eventually there becomes a point at which the population is no longer viable, and it will disappear down the plug hole. It will go down a vortex and become extinct. And that really is what we're worried about. And to come back to this idea of deleterious alleles and why they appear in the population, first of all, one of the parent animals needs to be carrying one of these deleterious alleles. And in this case, if we go back to our original analogy of the dam and of the sire, we're not just carrying A, B, C, D this time, we're carrying A, B, C, and X. And X is this gene that we don't want. It's a deleterious allele. But because it's carried alongside C, if it's what we call a recessive allele, it isn't physically seen in the animal on the ground. It's in there in its genes, but we don't see it. If we cross these animals in the same way we crossed before, this offspring has inherited A from the dam and X from the sire, but because it's in the presence of A, yes, it's a carrier for that gene, but we won't see it on the ground. It won't be expressed. And similarly, this progeny, it's inherited B from the dam, but also inherited X from the sire. Again, it won't be shown on the ground. It's there in its genes, but it's a carrier. It doesn't actually the trait doesn't manifest itself on the ground. If we now cross these two carriers, and both of these animals are carriers, in this case, this offspring has inherited B from the sire, an inherited X from the dam, again, it's a carrier. It hasn't got 2 copies. But this is the worrying situation, in that this progeny has inherited 2 copies of the deleterious allele. It's co-inherited the copy that's carried by the dam and the copy that's inherited from the sire. And this is exactly the mechanism by which the lethal fell pony syndrome is expressed. Okay, as an example, foal immunodeficiency syndrome. These animals won't survive. They'll be born, but they won't thrive, and they're unlikely to live for more than 24 hours. It's lethal after birth. So it's expressed because it's carrying 2 copies of that gene. And that's the mechanism for these recessive deleterious traits. And within Shetland ponies and within miniature ponies, there are known manifestations of these deleterious alleles. For example, dwarfism. We know that that's carried by Shetland ponies as well as miniatures. Also manifests itself in Friesians as well, actually. Another one is skeletal activism. If I was to look at that, certainly look at this picture, then in this one I would call that cow hock, but in the more extreme form, it's actually known as skeletal activism. And in terms of the inheritance of that, as we saw in the previous diagram, if neither of the animals are carrying the gene, then the offspring can't inherit it. They will be clear. But if one of the parent animals is a carrier, then by pure Mendelian inheritance, 50% of them will be clear and 50% of them will be carriers, but none of them will actually show it on the ground. Okay, it's the situation here where both are carriers that it becomes really disturbing, because 25% of the progeny will be clear, 50% will still be carriers, but 25% of the progeny will actually be affected by the trait because they will inherit 2 copies of the gene. So that's what we call the inheritance of autosomal recessive genes. You can get some traits that are dominant. In other words, you only need 1 copy. If any of you are breeding quarter horses, you'll know about polysaccharide storage problems, and that's a dominant trait. You only need to inherit 1 copy of the gene. So in other words, if it's carried and passed on, then it will be manifested. Polysaccharide storage myopathy. Now then, are all deleterious alleles a problem? And does nature do anything about it? Well, some of you might recognize these cattle. These cattle are extremely rare in the UK. They're from a population in the northeast of England of Chillingham cattle, and they are very, very close to wild cattle. And in fact, in genetic terms, these animals, each of the animals in these photographs, is almost a clone of each other. They are extremely highly inbred, but they're run as a wild herd with very little human interference. And even though they're highly inbred, they actually do extremely well. And the reason they do extremely well is that throughout history, for reasons that we can't fully explain, they've actually managed to purge deleterious mutations. We know about mutations. They're in the news lately. We all know about Omicron and COVID. We know that viruses can mutate. They mutate as they replicate. Well, so do the genes within horses. They mutate as they replicate. And in mutating, you can actually purge the genes. They don't get passed on. But we can't predict whether that will happen. And sometimes highly inbred animals can get rid of all the negative deleterious traits. And that's what we know has happened within Chillingham cattle. Unfortunately, we can't run a breeding program based on we'll increase the level of inbreeding in the fingers crossed hope that we'll get rid of all these deleterious traits, because the probability is it won't happen. And the reason is that we interfere. It's not like these animals, just turned out into the wild without veterinary intervention, without human intervention, and left to get on with it. We know as breeders, we make choices. When things go wrong, we call the vet, and actually random mating doesn't happen within our breeding programs. So we can't run a breeding program based on the fact that purging will take place and it will take care of all these deleterious traits. Okay, let's start looking at other breeds that we know a lot about in detail. And my expertise comes from understanding the Cleveland Bay stud book more than any other. And the Cleveland Bay stud book goes back to the 1700s. In fact, on the male side, we can trace it back to founders that are shared with the thoroughbred population. Those who know about thoroughbred history will know about the founders, and one of those being the Darley. We got the Darley Arabian and the Godolphin Arabian, but we've also got the Biley Turk, and he was a Turkoman horse. And on the male side, we now know that Cleveland Bays were descended from Turkoman horses. But the stud book itself wasn't started until the very late 1800s, about 1885. And what we do know was that the breed was already inbred at that time. But if we take it from the start of the 20th century, from 1900, and look at the pattern of inbreeding as breeders bred those horses right the way through the 20th century, it was already inbred at the turn of the century. It became more inbred, and then suddenly there was a drop, and that drop happened in 1920, just after the First World War. And the reason there's a drop, and we shouldn't get a drop within a ring-fence population, is because outside genetics got bought in. And in fact, they got bought in because they amalgamated two stud books. There was the Cleveland Bay horse stud book, and there was the Yorkshire coach horse stud book. And Yorkshire coach horses were first generation crosses of Cleveland Bays with thoroughbreds that gained favor in the late 1700s. They worked out that they could pull stagecoaches faster than purebred Cleveland Bay horses. They could get from York to London in under 24 hours because of the thoroughbred influence. And so they became very popular. But the Yorkshire coach horse stud book never produced more than 3 volumes. And they decided in 1920 to amalgamate the stud books. So what actually happened was we got a decrease in inbreeding. So that's slightly artificial. But then from about 1935, right the way through the Second World War, and carrying on right the way through to the millennium, inbreeding carried on increasing, and it went through 20% around about 1995 and was carrying on increasing. And it was at that point that I got involved with the Breed and the Breed Society. And we started to do more detailed analysis. And I'll talk later about how we actually moved that forward. But that was the picture of Cleveland Bay inbreeding up to the millennium. And that arrow there just shows you that trend, a very linear pattern. Once the amalgamation of the two stud books has happened, it's settled down, and then we've got a pretty linear rate of increase in inbreeding. It doesn't go backwards anymore, it just carries on increasing. So that's inbreeding. It's the relatedness of an animal to itself via its parents, via its sire, and via its dam. And that's the first concept I want you just to park up, because the second concept that I want to talk about is kinship. And that's about how related an animal is to any other animal in the population. That's about how related I am to my next door neighbor. It's about how related you are to your next door neighbor. So in other words, the relatedness of one animal to another that isn't directly its brother or its sister. So it's the relatedness of two individuals. And again, it's the probability that two randomly chosen genes at the same place on the DNA can be identical by descent through a common ancestor. But this isn't just in an individual animal, as in inbreeding, it's in 2 different animals. So full siblings were 25% inbred, but they're actually 50% related in terms of kinship. So kinship tends to be twice the inbreeding. If we go back to our diagram and have a look, we've got these animals which aren't inbred, they're completely unrelated. If we cross them, we said before that these are not inbred animals because the parents were unrelated, but because they're brother and sister, they have a kinship of 50%. So they're not inbred, but they have a kinship of 50%. If we were to now cross them, we've already seen that those 2 siblings, if we cross them, they're 25% inbred. What happens to the kinship? They've now got a 75% kinship. And if we carry on doing those crosses, we're now at 37% inbred. That's an example of how you can have inbreeding greater than 25%, all right, because we're doing crosses of inbred progeny already. And again, what will happen is this hypothetical cross will have an even greater kinship. So this is between 2 individuals. When we looked at inbreeding in those other breeds, it talked about the mean, the average. And what we can do is we can look at the relatedness of every animal in the population to every other animal in the population and average it out, and it will give us a mean kinship, the average kinship in the whole of the population. And that's actually a really good way of measuring how related all of your animals are to each other, and it's a really good indicator of the genetic health of your population. It's not the best indicator, but it's a good indicator. So in other words, if you've got a very high mean kinship, it's very likely that another measure, the effective population size of your population, is small. So a big mean kinship, in other words, your animals are all related to each other, it will tend to give you a very low effective population size. And I'll talk about that in a moment. So individuals with lots of relatives within the population will have a very high mean kinship, but equally the rarer animals, the ones that haven't got a lot of representation within the population, will have fewer relatives and will have a lower kinship. And low mean kinship animals are genetically the more important animals within our population. No matter how rare the population is, within that population, the ones with the higher mean kinship are carrying the more common genetics for the population. The ones with the lower mean kinship are carrying the rarer ones, the less common coloured Smarties, the less common coloured Sweets. They're the ones that are carrying those genetics. And that can actually help us prioritize animals for semen collection, for example. If we've got a finite pot of money and we want to target it, we would probably target it to making sure that the stallions with the lowest mean kinship were collected before the ones with the higher mean kinship, because they're the ones carrying the genetics, the alleles that are at most risk of being lost within the population. So, what do we understand by the term population size? Is it just the number of hooves we've got on the ground, the number of animals we've got? Because we can have an awful lot of animals, but if they're all homozygous, all carrying the same genetics, like in that population of Chillingham cattle that I showed you, does that actually mean the population is healthy? Well, not automatically. I talked about Chillingham cattle surviving because they randomly mate and they've not been managed much by human beings. That's because they mate in what we probably determine as close to an idealized way. Ideal populations are infinitely large. They undertake random mating. All individuals get a chance to contribute to the next generation and they contribute equally. That doesn't tend to happen in equine breeding, does it? That we don't have overlapping generations. Well, if you ever tried to sort out a stud book, we know it's full of overlapping generations. We know that real populations are restricted in size, and in horses, we know that we don't keep as many stallions as we do keep mares. that we tend to geld an awful lot of the male stock, that there's unequal sex ratios, there are unequal family sizes, that human selection is prevalent right through our breeding programs, and that they're wrapped up with overlapping generations. So our populations aren't ideal populations, and what we tend to need to do as geneticists is actually to look at what we call effective population size, and it is above mean kinship. It's the most important parameter in population genetics and conservation biology, or certainly one of the most important parameters, and it actually gives us a translation between the number of hooves we've got walking around on the ground and the actual genetic diversity that we've got, because the effective population size is the size of an idealized population that would be losing genetic diversity at the same rate as what we've actually got. And we can measure effective population size in a number of ways. Historically, it used to be measured from the number of breeding males and breeding females. In other words, the animals that are contributing to the next generation. But more importantly, what we now know is that's a bit of a corruption, and that it ought to be measured from the rate that inbreeding increases in each generation. And that doesn't mean the rate that it increases year on year on year, but with each generation. And in most equine breeds, the generation interval is actually about 10 years. That might sound quite a lot, because we know we can breed from a mare at 3 years old, but the average tends to be across a whole stud book somewhere around about 9 or 10 years. And so we measure effective population size over complete generations, and it's the rate that inbreeding increases with every generation. And that's the only the true and proper way to record effective population size, is by knowing how much your inbreeding is increasing. There are some rules that have been formulated by geneticists over the year, and that's one worth bearing in mind is the 5500 rule. And that says that we need an effective population size of 50, an effective population size determined from inbreeding of 50 for short-term survival of the population. And if we've got an effective population size of less than that, the population isn't viable. And that tends to be because the natural purging of deleterious alleles is overtaken by the increase in their appearance because of the increase in inbreeding. So actually we get more and more deleterious alleles, we get more and more lethal equivalents appearing, and that in turn is making the breed less viable, less fertile, and less successful reproduction. And then an effective population size of more than 500 is needed for the long-term survival. And that tends to be measured, all right, now we know slightly incorrectly, from the number of parents, the males and females that contributing to future generations. And we need that figure of 50 to make sure that we don't put our breeds into what we call inbreeding depression. And interestingly, when governments report to an agricultural organization of the United Nations, the target that's actually set is that we should be maintaining our livestock breeds at or above, and preferably above, an effective population size of 50. That's the baseline. And the target is 500 for long-term survival. And in fact, there's now an argument that we should change the 5500 rule to a 100-1000 rule, but that's another debate. And if you want to look up the reference to that, the reference is Franklin from 1980, who first came up with that 5500 rule. So we know a little bit now about the problems that we have in closed stud books from inbreeding and the associated increase in kinships and decrease in effective population size. How can we actually measure them if all we've got is stud book data? Well, there are various bits of software now. Computers have helped us an awful lot. And I'd like to mention one that's been formulated really for the Food and Agricultural Organization of the United Nations. It's a piece of software called PopRep, and that's what I do most of my genetic analysis with. And that will give us a detailed report of population statistics, of the size of our population, of our number of foals we're getting year on year on year, and their makeup. Have a look at what animals have contributed to the population most, the animals that have contributed least, and it will also measure effective population size for us. So there is useful software out there with which we can determine rates of increase in inbreeding and changes in effective population size, and we can monitor our populations. And if we have a look at the breeds that we're interested in, and we're particularly interested tonight in the Shetland pony, in terms of effective population size, this is from the UK Shetland pony population. And this is only from a very small sample size, but it's done through molecular methods. And in sampling 27 horses, they found that there was a molecular effective population size of 365. So you can see that's well in excess of the 50, but it doesn't reach the 500. And if we look at the heterozygosity, in other words, how many copies of different alleles it's carrying, then we can actually have a look and look at the heterozygosity in the breed that we look at. We're around about 26% across the board there. In terms of inbreeding in the UK population, they're finding inbreeding a minimum of 10%, a maximum of 37%. And that was higher than from the Netherlands population, if you remember. And the average there is 18%. So that's what they're finding in the UK population of Shetland ponies. If people want the reference for that work, I can provide it later. If we look at the size of our population, if we've got a population that is an effective population size of 100, then it will increase in inbreeding by 0.5% per generation and lose 0.5% of its diversity per generation. If we halve the effective population size down to 50, we can see that we lose 1% per generation. And that's why the Food and Agricultural Organization of the United Nations chose that red line in the sand. They don't think breeds should be losing that at all. And certainly that if you're losing 1% per generation, you become a non-viable population. If we halve the effective population size again, down to 25, we can see if we halve on the left-hand side, we double the right-hand side. So if we halve it down to 25, we're now losing 2% of a generation. Halve it again, we lose 4%. Halve it again down to an effective population size of 6, we're losing 8% of our diversity per generation. And if we don't start off with a lot of diversity, you haven't got much to lose until you've lost everything. Just bear in mind these figures 12 and 25 because they relate to what happened within Cleveland Bays. You'll see that Cleveland Bay's effective population size dropped down below 20 and it was losing more than 2% per generation. In fact, at times it was approaching 4% per generation. If we look at sex ratio, an unbalanced sex ratio and the effect that that has on effective population size as well. If we take a population or a census population of 100 animals, okay, and it's 50-50, males and females, then the effective population size remains at 100, where we've got that balanced sex ratio. And just as in the previous diagram, we're losing 0.5% of diversity per generation. But if we start to skew things, we still keep 100 animals, but this time we've got 25 stallions and 75 mares. We've now got an effective population size of only 75, and we're losing 0.7%. Sorry, we're losing 7% of diversity per generation. All right, 0.7%. If we now go and change the ratio again, still got 100 animals, we've got 90 mares and 10 stallions. The effective population size has dropped to 36, and we're now losing 1.4% per generation. And if we carry on that exercise, sorry, I just moved forward, I just moved back. If we go down and we now got only 2 stallions and 98 mares, we can see that the effective population size has dropped to 8, and we're now losing 6.5% per generation, and that we're well below that target from the Food and Agriculture Organization. And to be quite honest, a ratio of 2 to 98, not that unusual within equine breeding. So just by the nature of livestock breeding and the fact that we keep fewer males intact than we do breeding mares, it actually brings its own problems. So in managing our pedigree populations, we do have choices. What we could do is we could divide up what we've got left into subpopulations, and we could keep them apart. And to a certain extent, that's happened in Cleveland Bays, and it's happened in Suffolk horses, because with Cleveland Bays, back in the late 1800s, a lot were exported from the UK to America and Canada. And they've also gone to Pakistan and to Japan. And there hasn't been until relatively recently, there hasn't been an awful lot of genetic exchange back the other way. So the populations have been kept separate and they have developed separately. And that actually helps to increase diversity. Certainly within the Suffolk horse population, they again were exported to the USA and Canada as working horses and for breeding purposes. And there's been almost no exchange back the other way. And the Suffolk horse in North America is actually fairly different now, both physically and genetically, from the UK Suffolk horse population. And that can be quite a good thing in terms of the doomsday scenario, in that there is a genetic resource on the other side of the Atlantic that could be tapped into if the UK population became so compromised that it became unviable. Another choice would be random mating, and that's again is what's happened with the Chillingham cattle. But I already said that random mating doesn't happen in the real world. We make choices as breeders. We call the vet when things go wrong. And you know, what I choose as being a Cleveland Bay that I quite like, my next door neighbor might not like at all, and so wouldn't make the same breeding choices. So we tend not to breed randomly. The other choice that we've got is to manage through control of inbreeding. And research from zoos tells us that that is probably the best strategy, but better than that, actually managing inbreeding through controlling mean kinship is gold standard. And that is the best way at the moment that we can manage populations that have already got genetic diversity that's compromised. There is another system called Optimum genetic contributions, but that you can only use in populations that aren't genetically compromised, because you then start scoring on physical traits as well as genetic traits. And if you're not careful, you end up pouring genetic diversity that you can't afford to throw away down the plug hole if you adopt those strategies with rare breeds. So managing of inbreeding. And we do that through minimizing mean kinship. And what that can do is it reduces the rate of increase in inbreeding from one generation to the next. It stops us bringing animals with the rarest genetics directly into mating with those carrying the more common genes. And it maximizes the amount of genetic diversity we retain in future generations. It also helps us to identify the most important of our rare animals to breed from, both be they males or females, so that we can go away and we can do genomic collection, we can do semen collection, or we can set up incentive schemes to make sure that the rarest animals do get bred from. In Cleveland Bay terms, most of the work has been done looking at pedigrees and stud books, but we have to ask ourselves, is that relevant when we do know what the horse genome is? You know, when we have got genomics, when we can do parentage assignment through microsatellites, and we're not far off moving forward to, as routine, using different panels of SNPs, SNPs, single nucleotide polymorphisms. What about genomics? Is there still a place for a stud book and the stud book record? Well, there's been analysis in a comparative analysis with thoroughbreds, a paper written in Australia that looked at the Australian thoroughbred population. And when they compare genomic data with pedigree data, what they found is that where you've got a very deep and robust pedigree, there's actually a really, really good correlation between pedigree-based inbreeding and genomic-based inbreeding. We saw that in the example that I gave you of the Frisians, where they have got an accurate stud book, and it is giving you a reasonably good comparison between genomic measures and pedigree-based measures. So yes, there is a place for pedigree. And the thing is that as breed societies, most of us have already got really robust stud books that go back lots of generations. So whilst we might have a limited parentage testing database, we've probably got a more complete stud book. And as long as it's robust and complete and error-free, then it is still, and going forward, it will still be a valuable tool for helping us manage these breeds. But we can't breed by stats and numbers alone and by knowledge of genetics alone. As breeders, and first and foremost, you know, I am a horse breeder, not just a biologist. We choose with the breeder's eye. We look at the horse's phenotype. You know, has it got a leg at each corner? How does it move? Is it cow-hocked? What's its conformation? As well as looking at and understanding its genetics. You know, what's the makeup of the mare? What's the makeup of the stallion? What common founders do they share? What's the inbreeding of a potential mating going to be? And what we need to do is to make sure we can provide breeders with tools where they can use their breeders' knowledge, but also have more understanding of the genetics, greater than they would gather from just shuffling around a piece of a pedigree on pieces of paper on the kitchen table. And there's a really good reason for that. And I learned this very early on, is when you go through stud books and you start to write out pedigrees, well, in a lot of passports nowadays, we'll easily have a 5-generation pedigree. And a 5-generation pedigree there, I'm just going to minimize and move out of the way my little icon of myself on the screen. In a 5-generation pedigree, there will be 32 animals in that 5th generation. If we dig a bit deeper and shuffle around, it is still just about possible on one piece of paper to print out an 8-generation pedigree, but we're using a very small font now for the great progeny, and the fact that 8-generation level, there are 256 animals. And for most people writing down on A4 pieces of paper, they've probably moved on to a number of pieces of paper that they're sticking together with adhesive tape. If we go to 10 generations, we've probably now moved off shuffling those pieces of paper around on the kitchen table. We've now moved on to the kitchen floor to make sure there's enough room, because we're now talking about 1,000 animals in that 10th generation. Double that up and go to 20 generations. We're now not talking about thousands of animals. We're talking about over 1 million animals. We're talking about going to a football stadium with those pieces of paper and spreading it out all over a football pitch. And you're not going to do that in your lifetime. That's why we start to need computers to understand deep inbreeding and relatedness within a pedigree. But I told you before that the Cleveland-based stud book goes back well over 32 generations. Well, what happens at that sort of a level? Well, if we go back 32 generations, we've now got not millions of animals, we've got billions, over 4 billion animals in that pedigree. And that's for each of the animals that are alive in the population now. We understand the pedigree, if it's complete, to that number of animals. Well, hang on a minute. At this moment in time, the world horse population is estimated at 15.8 million animals. So if Cleveland Bay horses were completely non-related, in other words, if their inbreeding was 0, that would be impossible, because there are only at this moment in time 15.8 million animals in the world. In other words, any pedigree that is deeper than 25 generations by default has to be inbred. There aren't enough animals to form the founders of the pedigree. And if we look back in most pedigrees, there are common founders on both sides. There simply aren't enough horses in the world for our populations to be unrelated and uninbred. It is important we look back down these generations. This comes from a website called breedingfordiversity.com, and I'll refer you back to that later. But this is looking at actually kinship in dogs, and it's looking at the difference between a 7-generation pedigree and going back the full depth of the pedigree, and in the case of these dogs, approaching 30 generations. And we can see what the pedigree looks like at only 7 generations deep. It looks like it's made up of a structure here with just these few types or groups of clusters of animals. But if we reveal the whole pedigree, we can see that there's a lot more fine detail and a lot more founders contributed to it. And it's giving us a true population structure from which we can assess proper inbreeding. So it is important we go back all of the generations in a stud book. And that means that if we've got a stud book, it is worth digitizing the whole lot. And once we've got a stud book, we can start to use it in detail as another tool to give to breeders. And this is a breeding program that I adapted using computer software that was available in the 1990s from a population that came, a piece of software that came from the American Zoological Association called Single Population Animal Record Keeping System. And that is still to this day in an online form used in zoos throughout the world. And it's used to look at kinship and inbreeding within captive wildlife populations. And I've adapted it certainly for the Cleveland Bay breed, and now more recently for 2 of our other equine rare breeds in this country, and that figure's still expanding, the Shetland pony and the Suffolk horse, so that individual breeders can have information about their mares and how the crosses with all of the available stallions would stack up. And I'll look at it in a little bit more detail for you and show you how the system works. It's developed over the years into a traffic light scheme, which simplifies things. And you can see that for this mare, Kenley, Nova Scotia, who belongs to a friend of mine. This is the table from last year. We're just producing them for the 2022 breeding season, and they do change each season as the population changes, as animals are foaled, as animals, for example, colts get gelded and couldn't contribute to the next generation, and also as animals die without their genomic products being frozen. But we can see that there's a color coding here. We've got green matings, yellow matings, orange matings, and red matings. And the color is important because we try to encourage these green matings. These are matings between animals of very similar mean kinship. And if you remember, that means animals that are carrying similar genetics. We don't want to mix the animals that are carrying the rarest and the most common genes, because that will leave problems in the future. It will reduce the number of options for future generations. It will also, the green matings give animals of lower co-ancestry. In other words, the theoretical inbreeding coefficient of the animal is lower than the mean kinship of the mare. And you might say, well, why isn't it just lower than the average for the breed? The reason is that for a lot of these mares that are quite highly inbred already, that would leave very few green matings. So we, over the years, have developed the scheme and found that it's very effective if we actually take the mean kinship of the mare as the target level for inbreeding in the foal and actually pitch it at that. Matings giving an inbreeding coefficient of the foal lower than the average for the population would be ideal, but not everybody can do that. And for whatever reason, whether it's for geography or simply for some mares, there aren't a lot of green matings. We've got best of the rest matings. These are the ones that we encourage people to do if they can't do a green mating. And these still mate, and this is the important bit, they're still mating animals of similar mean kinship, because we really don't want to mix the animals with the rarest and the more common genes. These might be producing animals of higher inbreeding coefficient than the mean kinship of the mare, but they are lower than a threshold that we set for the breed. And if we can't do a green mating, these are preferred, and they're still advantageous. We want these animals in the population, the progeny of these matings for future mating. Now we're getting to a level of matings we would rather breeders didn't do. And the reason is because we're now bringing the rare genes and the common genes together in a way that makes them irrevocably, inseparable. Because we not only do we increase the frequency of the least common genes, but we're also at the same time increasing the frequency of the more common genes. And these are matings that produce co-ancestry of higher inbreeding coefficient than either of the parent animals. But it's still lower than that threshold that we set for the breed. So we really would rather people didn't do these, but we're not going to penalize them if they do. We would rather they didn't. And as I say, we're mixing rare and common genetics, and that's not good for the long-term survival of the breed. Then we get to the red matings, and these are matings that are very highly inbred. And we've already seen the problems that highly inbred matings bring. The increase in homozygosity within the breed and the increase in deleterious alleles. So we've got animals of very different mean kinship, and we've now got animals of higher inbreeding than the threshold. That's set at 24% for Cleveland Bays. We've set it at 18% for Eriskays, and we've set it at 12.5% for Suffolk horses. For all the reasons that I've explained. So with that mating scheme, which has been in place with Cleveland Bays since the very late 1990s, we started to tinker with it in about 1996 as a result of some breed conferences. We put it formally in place in 2004. I've already shown you the graph where we looked at the increase in inbreeding between 1900 and the turn of the millennium. If we actually have a look at what's happened since we implemented that breeding scheme and we made those sheets available to breeders, we can see that the progeny that's been registered since, yes, inbreeding is still increasing, but it's not increasing at such a rate as it was before. We've slowed things down. And in turn, what that's done is it's really helped us with effective population size. Because back at the turn of the millennium in the year 2000, the effective population size was down below 20. And if you remember, we were losing genetic diversity at approaching 6% per generation. We implemented the scheme in 2004 formally. And since that time, as more and more breeders have taken the scheme on and adopted it, and as we've been able to secure funding and make incentives available for breeders to preferably do green matings, we've actually seen things start to improve quite dramatically. And we went through that magic 50. You remember the 5,500 rule? We passed 50 in about the year 2014-15, and it's carried on increasing. And within the last year, we've seen it go from about 140 in the latest year to in excess of 200. And this is measured from the rate of increase in inbreeding. So it's a true representation. So in the Cleveland Bay population size, where we've managed to run this scheme for in excess of 16 years now, it's been extremely successful in reducing the loss of genetic diversity and actually retaining genetic diversity for passing on into future generations. And we've measured that by the increase in effective population size. And that in turn has been reported back. It's been reported back to government, and it's reported onto the Food and Agricultural Organization of the United Nations. And governments have to, you know, there are targets set. The ACI target is for, ACI target number 13 is for governments to improve the retention of genetic diversity within its livestock. So we're helping governments meet targets. In terms of what we managed to do, we've got a rate of increase in inbreeding that's been reduced, and that's helped us to bring slow it down. It'll never be zero unless you start to bring animals in from outside the ring fence of the stud book. If you do try to bring inbreeding down quickly, then what actually happens within your population, if you bring those rare and common genes together, you can create very relatively low inbred progeny. You'll do it in one generation. You might be able to do it in the second generation, but you won't be able to do it in the third, fourth, and fifth generation. So what actually happens, and this is known from work with captive wildlife animals where the generation interval is much shorter, that this is why we manage through mean kinship and not directly by minimizing inbreeding itself. Because if you just try to target low inbreeding coefficients, then you will compromise yourselves in third, fourth, fifth generations. And those of you who breed dogs, especially if you're breeding dogs within the United Kingdom, will know that the UK Kennel Club has implemented a sophisticated piece of software now that does predict inbreeding coefficients in prospective matings, but taking no consideration of mean kinship. They've missed out a vital part of the science. And unfortunately, the generation length within canines is a lot less than equines. They, unless they do something about it, are going to find they've shot themselves in the foot, and they have a problem in the next few generations very, very quickly in real terms. That message needs to get across. So we need to make sure we don't do that, because as I say, and it's a classic example is what the UK Kennel Club is doing right at this moment in time, is it's going to limit options for the future. So just to recap, when we select these matings, we need to try to breed from as many males and females as possible. That's why we provide the information to the mare owners, showing all of the possible stallions, not just the good ones. We show them everything, not just the green matings. We try to encourage people to rear colts and keep them long enough so that they can be licensed, providing there's no reason for not to license them and use them. That helps to maximize effective population size. And we try to discourage the overuse of a small cluster of stallions, and that can be difficult. The stallion in the photograph of a Suffolk horse called Craig Howe Jensen, who about 3 years ago came into the hands of a very keen breeder who understood sparks, but didn't realize that his overzealous marketing of his stallion would mean that and he got it into the national press and into the equine press as well, how good his stallion was. He was out competing it. He was actually jumping this stallion, which is unusual for a working horse, for a heavy horse. And in the following year, he was so successful that there were 11 foals by that stallion out of a foal crop of 66. So a substantial proportion of 1 year's foal crop was by that one stallion. And that had a knock-on effect in reducing effective population size. So we need to be a little bit careful. But in terms of breeding for the future, we need to think about that toolbox. And breeding schemes and advisory schemes like Sparks, you know, they can be a help to breeders, but they're not the be all and the end all. They leave the breeder in control. That's really important. But what they do is they mean that we can avoid those red, highly damaging matings. That in turn maximizes the retention of diversity in future generations. And we can encourage the mare owners of those really low banded mares, we can flag up with them that their mare is really important from the breed and actually try and put incentives in place so that they can still be actively breeding from those mares. I put this slide up because some of you that follow the Suffolk breed will know that a couple of years ago at our premier artificial insemination centre in the UK, they've now got the technology to sex semen. They can collect from a stallion. It's not frozen semen, this is from chilled semen. They can, using technology from Cogent, they can do some electrophoresis and separate the semen out into semen that is statistically going to bring about a filly foal. It's sex semen. And they successfully, using sparks to choose the mare and the stallion, managed to produce a filly foal. And if we could extend that scheme to make sure that we are producing green matings that are going to increase the number of filly foals on the ground, that has to be a really good thing for our breed. So this sort of technology can help, and Sparks is part of that program. But what we do know is that going forward, uncoordinated breeding, as we've seen with the Cleveland Bay and with other breeds, it leads to it can lead to an unsustainable increase in inbreeding. That has a direct influence, a negative influence on effective population size. I think from our breed point of view, we need to stay up to date and understanding our breed. And there are tools there, PopRep, for example, to understand our stud books and see what's going on. There are also now commercial tools that are starting to be developed. I'm working with Sparks in a non-professional way, but there's now a stud book management program called Cloudlines that has started to adopt exactly the same science that Sparks does to make these breed advisory schemes. But what we need to bear in mind behind it all, certainly for those countries that are still within the EU bloc, I know Britain, the UK have left, but we still got animal breeding legislation that's based on the UK EU zootechnical regulations that firmly put breeding decisions in the hands of breeders. Breed societies cannot dictate what breeders do. They can provide advice, but a scheme like Sparks helps provide that advice, and use of software like PopRep means that breed societies can monitor their overall populations and see what effect individual breeders are having. I mentioned earlier on this website, breedingfordiversity.com. It's by a geneticist called Pieter Oelihek. Your pronunciation of that would probably be much better than mine, but he goes into some detail if you want to dig into the background of why mean kinship is important in our breeding programs. It's part of his 4 steps for conservation, whether they're captive wildlife populations or canine populations or equine populations. So I'd refer you to that website for some background on the science in a relatively digestible form. In terms of the Cleveland Bays and validating what we've done, in conjunction with Nottingham Trent University and the Rare Breed Survival Trust, I've been co-author of a recent peer-reviewed published paper that was published. It's open access in Ecology and Evolution, which is a Wiley journal, and that was published last October. It looks at and validates 16 years of breed management within the Cleveland Bay breed and looks at how we've managed to increase effective population size. I think it was important that it was independently peer reviewed. It wasn't just Andy Dell saying that it was working. It's now been validated by other independent researchers. That's available online. Yeah, that's the web address, but if you just type in 16 years of breed management Cleveland Bays, you'll hit that as your first hit on a Google search. It's quite a detailed paper going into the background science as well, but that's chapter on verse and tells you about the scheme and about how it's worked and the science behind it. So there we are, folks. I hope you found that interesting. I'm sure it will have provoked some more questions. I know we took one or two during the talk. I'd personally like to thank Linnea and the Shetland Pony Society of Sweden, and also I know the Federation of Swedish Horse Breeders has become involved in sponsoring this and making this available. I think it's really good that I'm looking now, we've got nearly 120 people participating in this webinar. So Linnea, thank you for inviting me to do this talk, and thanks for the opportunity of doing it. And more than happy to take questions, and hopefully we get some interesting questions coming in. I can see looks like there's some in the chat, but it's been hard for me to monitor the chat as we go along.
Thank you so much. It's been really, really interesting.
I'll just stop my screen share for the moment, and then we can see people's faces.
Absolutely. So we're actually up to, I think, almost 150 people at most.
Wow.
We have some questions that were a bit back, but how can the inbreeding coefficient be lower than 0?
Inbreeding can't be lower than 0. What happens in some research is if the pedigree isn't complete, or if outside genetics have been brought in and a stud book analysis is done, it can end up in stupid minus figures. They're completely unreliable and you shouldn't rely on them. In real terms, inbreeding can't be less than 0.
Okay. And we also had a question about the microsatellites. Are there any markers that are more important not to have on both sire and dam's side?
Right, in terms of microsatellites, there are an awful lot more than 17 microsatellites in the horse's genome, but that panel of 17 has been chosen specifically because of their ability to define males and the parents. So in the ones that you were looking at, they're equally important. Down at the bottom of that Lex 33 is sex linked. It's on the X chromosome. So out of them, if you had to choose one that is least important and you could probably do away with it, it would be the one that's sex linked because it's not replicated on both sides of the pedigree. But I think fairly soon, I mean, there are published papers. Paul Flynn at Weatherby's Incorporated in Ireland at their lab in Kildare has been co-author of a paper, and his immediate supervisor, Rumi Morin, is part of the International Society of Animal Genetics panel that are working on what markers they're going to use out of the next generation SNPs panel, and we'll be moving over to potentially screening 50, 60, 70,000 markers, of which they will choose probably hundreds for parentage verification. And again, they'll be equally important. So
from what I understand, there's only a few microsatellites that you use for parentage. So I mean, you need a lot more than that to look for common kinship and how to use them for breeding.
A long time ago, at the start of this study, I tried to use the microsatellite data set that we had for Cleveland Bays, which at the time consisted of samples from 500 animals. It's up to over 1,000 now. But because there's such a restricted panel of markers, the results you get back from trying to do computer analysis of that set is not particularly meaningful when you try to look for population structure. But if we were to move across to SNPs markers, where we're talking about thousands of markers, there are plenty of papers, of peer-reviewed papers in the literature now, which give us detailed analysis, and they're not looking at that restricted panel of microsatellite markers. We've moved on to the next generation of markers now, and they can tell us things. For example, with the Eriskay ponies now, they're starting to find ancient markers that are detectable in current Eriskay ponies and in ancient ponies, and they are starting to work out that Eriskay ponies certainly are a breed in their own right genetically, not just a variant of the West Highland pony, as had been said, or of Highland ponies. And there are specific Eriskay pony traits they detected within that panel of SNPs markers. Okay,
we have a question from a breeder of Arabian horses.
Sure.
And they have the recessive genes for CA and SCID. They have mandatory testing to be able to register the horse. How worried would you be to use a carrier on a non-carrier looking at the whole picture of the breed?
If I can come away from the Arabian situation to something that I'm more familiar with and talk about how, again, a recessive allele can be tested for within, let's say, the fell pony, where we've got foal immunodeficiency syndrome, and where it's straightforward inheritance. We know full well that they, if one of the animals is carrying that, it could be passed across into the next generation and be silent and not expressed. If we have two carriers, then there is a 25% chance it's going to be expressed within the progeny, and certainly for foal immunodeficiency syndrome. In other words, 25% of the animals that are bred are not going to survive longer than 24 hours. So it's really important, and that trait has carried across because of indiscriminate breeding. It's carried across into the Dale's pony, and the Dale's pony is now, because we can test for that deleterious allele, has now instigated a program where they will not license a stallion that is a carrier. So in other words, none of their stallions will carry it. So if they start screening for it within the dams as well, we'll be able to get rid of it from the breed, but it shouldn't have been in the first place. If we take it from that lethal situation into a cosmetic situation, I can again relate to that in the Cleveland Bay breed. The bay color of Cleveland Bays, of any bay horse, is based on the agouti gene. That's what gives us this bay black color. It's a dilution, and there is a recessive gene involved in that, the chestnut gene. Chestnut gene is recessive, and again, from indiscriminate breeding that wasn't properly reported within the stud book, the Cleveland Bay has inherited that recessive chestnut gene, and every now and again we do get chestnut foals. They tend to be, rather than bright chestnut, they tend to be liver chestnut. And the chestnut gene, they identified and found that they could test for it very early on in the 1990s, and it had been suggested that to get rid of the chestnut gene from the breed, we should just screen all stallions, and any that were carrying it should be banned. Well, the problem is that whilst you might be able to do that in a breed that's really numerous and has a lot of genetic diversity, in a rare breed, you haven't got that luxury, because if you suddenly turn around and said, well, we're not going to breed from that stallion because it's carrying the chestnut, even though it's not chestnut itself, it's carrying that chestnut gene, you would throw away not just the chestnut genetics that it's carrying, but you would throw away all of the other diversity as well. In other words, all the other different colored sweets, and they'd go down the bath plug with the bath water. So whilst we can test for these deleterious traits, and as we know more about the horse genome, and especially with the coming of SNPs testing and the bead chip, which is how they measure these SNPs and profile them, we will be able to detect more and more of these deleterious alleles. I think each breed is going to be a case in itself as to whether they should screen, and Cleveland Bay's decided they shouldn't screen for the chestnut gene because it was cosmetic and not a physically deleterial lethal trait like the foal immunodeficiency syndrome. And again, I don't understand an awful lot because it's not my speciality about that particular deleterious trait in the Arabian population, but it needs to be seen in context to the impact that it's having on the breed as to whether they should put a screening program in place if it's not in place already. And obviously, the more we know about the genetics of it, the better. What we do know is that more and more of these deleterious traits that we understand, and we know we now identified going on for 46,000 of them, that a lot of them are lethal at embryonic level. And if we can detect them, and if we have got the genetic diversity where we have the luxury of being able to screen for them and then have a breeding program that starts to exclude them, then that's got to be for the benefit of breeds, because it will increase fertility.
Okay, excellent. You mentioned that there are 15 million horses estimated in the world. Is that 15 million currently? And you said because the sheet said 58 million. So that's
yeah, it's of the order of, I think the current estimate is 58.5 million horses is the world population. So it's of that order. And I think I just picked the 50 million as just a comparative number when I was looking at the number of descendants there would be in that 20th generation. Yeah.
Okay. So we have a longer question here. I have always learned that you cannot find parents by DNA if you don't have suggestions of parents to test against. But lately I've heard that in the UK or Ireland that is possible. That is, you could find parents to an unknown foal amongst all the horses in the database. Is this true? Is it due to the amount of markers used?
Right. Okay. So if you've got a horse that looks like a Cleveland Bay, can we pull its hair, send it off to the lab, get a DNA profile, and then compare it with the DNA database that we've got and go, what's the likelihood, can we identify the parents? And in a small population like Cleveland Bays, yeah, we have done that. It is possible. It needs computer sifting, but the problem is that at the moment we're only testing for a panel of 17 markers, and what's the chance of that makeup within 17 markers if you've got all of the parentage testing results from all the horses that have been tested in the world, of finding that replicated and actually not being those parents? Well, statistically there is a chance of it being wrong. As we move across to the panel of SNPs markers, where we're not using just 17 markers, but we're using potentially 50,000 markers, then we move to a situation where the chance of it being replicated across a correct and an incorrect set of parents is much reduced. So we are moving towards a situation where we can do that. We're not necessarily there yet. So it might be possible.
Okay.
If you've got access to the database.
Mm-hmm.
Yeah.
How has the Cleveland Bay population been affected when it comes to conformation and performance after the breeding program?
Okay. I'm not sure who asked that question. That might be a Cleveland Bay breeder that I didn't share the link. Right. In terms of conformation, it's important that you realize that breeding decisions are made up of phenotypic decisions and the genetic ones, and the Sparks breeding program is based on providing the genetic information, and we stress every year it's up to the individual breeder to use their breeder's eye to determine the breeding. Now, there are some breeders that will say that the Cleveland Bay is not of the type that it used to be years ago. Some people believe it's the problem of the breeding program. Others think that's a complete and utter load of nonsense, and there is no evidence either way. What's important is that we provide education. What we unfortunately can't do is we cannot implement a program like, for example, the Irish draft have done of linear profiling, where they physically go out and measure and score the conformation of their animals. In this linear profiling system, we get a complete score for the compatibility of a mare and a stallion, to look at how well they complement each other in phenotypic terms. And if we were to do that, we'd be doing something very similar to screening for the chestnut gene, in that we haven't got the genetic diversity to have that luxury of providing a phenotypic score as well as a genetic score. I don't know if we've got any Icelandic horse breeders with us, but they have a system based on both genetics and performance in terms of Icelandic pony breeding. Well, they've got a lot of diversity with, I think I get negative marks if I call them Icelandic ponies. They're Icelandic horses. They've got this scoring system, and they've got something much more akin to what we call optimum genetic contributions, which is the gold standard if you've got a lot of genetic diversity remaining already. So there is anecdotal evidence from some breeders that the breeding program has not improved the phenotype of the breed. There are others that say it's up to breeders to maintain the phenotype, taking on board the genetic data that we're giving them. So yes, phenotype is just as important as genotype, but the study and the paper that I've written is looking at genotype and how much genetic diversity we've retained to pass on to the next generations and future generations. It hasn't looked at the phenotypic aspect of the breeding.
Yeah, we have one last question, and that is from a breeder who's saying she's always been searching for individuals who are not related. But if this is not good for a genetic variation, how should you pick the individuals? Or do you have to know their genome to breed from that thought?
No, you don't need to know the full genome. Again, going back to what I was talking about, is we're looking for animals that are not related, but equally, we're looking for animals of similar kinship. So in other words, they are going to be slightly related, but not so related that it's going to cause a major increase in inbreeding. So if you've got a stud book that you've got digitized, even if you haven't got a breed management program that's as sophisticated as the Sparks one for Cleveland Bays and the Suffolks and the Eriskays, you can still identify animals of similar mean kinship that will give you animals of lower inbreeding coefficient than the rest of the breed. And so it is possible, if you've got a digitized stud book, to do exactly what you're trying to do, which is to look for animals that are not related. But it's a measure of what did we call not related, because the biggest outcross isn't necessarily the best thing for the breed in the future.
So how should you pick the individuals to breed from then? If you want
what I'm saying is, if you have a breed society that's got a digitized stud book, then it is, once it's digitized, it's possible to do the analysis that will give you the kinship figures for the breeding. If you haven't got the sophisticated traffic light program, you can still pick from the figures that it's giving you. If your pedigree is that sophisticated that you can't work this out from shuffling the pieces of paper around on the kitchen table, then you need to go to computer analysis of the stud book. You don't have to go down the route of having a genetic profile from DNA analysis of all of your animals. You can do it from pedigree analysis, but it's pedigree analysis that's carried out by computer analysis.
Okay, then. Thank you so much. It's been a very enlightening 2 hours, and I hope everyone's got their questions answered. And yeah, thank you so much for participating, and thank you to everyone who also have been listening. It's been a very, very broad, like different breeds participating, and lots of different breeds. And it's been very, very interesting. And that's the comment as well in the chat. So thank you very much.
Thanks for that. And what I will say is that if there are people that come up with questions that they wish they'd asked and they didn't have a chance to ask during the seminar, if they perhaps feed them back through via you, then you've got my email contact, and I can try and address them for people. I know the seminar's been recorded. I think it's really good that we can actually make it more widely available by sharing it on YouTube as well.
Definitely.
Thanks for doing that.
And if anyone wants to ask questions, they can email info@shetlandspony.se, and then we will try and route the questions to you.
Yeah, and if I can't answer them, I'll try and find someone that can.
Yeah, thank you very much.
In the right direction. Okay, thanks for the invitation. Thanks everybody for participating. And thanks for those that I think we've still got nearly 100 people stuck it right out to the end.
Very impressive.
Thanks a lot.
Thank you.
Good night, everyone.
Good night. Bye.